Good morning everyone. This is Raghu, Assistant Professor of Biotechnology from SR and BGNR Government College, Khammam. Today I am going to discuss few topics of molecular biology and are also called as basic experiments or topics involved in molecular biology. Those are blotting techniques, DNA fingerprinting and polymerase chain reaction, which is also called as PCR. While coming into the blotting techniques, there are three major blotting techniques which are called as the basic blotting techniques and heart of molecular biology. Those are southern blotting, northern blotting and western blotting. Southern blotting is widely used for the identification of DNA of interest, whereas northern blotting it is used for the identification of RNA of interest, mRNA of interest or a piece of RNA from the entire RNA. The other one is western blotting, which is different from southern and northern blotting because it is used for the identification of proteins, that is antigenic proteins or any other proteins. So these are the three molecular biology techniques. So the first one is southern blotting. Southern blotting, as I mentioned, it is used for the identification of DNA fragment of interest, where later it can be used for sequencing. The first thing is the entire genome of DNA is taken. You can watch it in the left side, and it is subjected to restriction endonucleases, where you got double-stranded DNA fragments. And these double-stranded DNA fragments are denatured to single-stranded DNA fragments. Here in the diagram it is not shown. And once the double-stranded DNA molecules converted into single-stranded DNA fragments, then it is allowed for agarose gel electrophoresis. In agarose gel electrophoresis, the DNA fragments are sorted based on the charges to mass ratio. The larger fragments they move slower in the gel, whereas the smaller fragments they move faster in the gel. So these different sized DNA fragments which are obtained after denaturation as well as restriction endonuclease treatment are allowed for agarose gel electrophoresis, are sorted. After sorting, they are transferred onto nitrocellulose membrane. So agarose gel is stacked with nitrocellulose membrane and an alkali is used for the transfer of these DNA fragments from agarose gel onto nitrocellulose paper. Here, for the transfer of the bands from agarose gel to nitrocellulose paper, in general, electroblotting is used, where the gel is stacked in between the nitrocellulose paper and electric mobility along with an alkali is used for the transfer of bands from gel to nitrocellulose membrane. After the transfer of transfer of bands onto nitrocellulose membrane, now our band of interest or DNA fragment of interest are identified with the help of probes. Probes are short nucleotide sequences which are labeled. Generally, these probes are of two types, either enzyme labeled or radio labeled. If you are using a probe which is enzyme labeled, then you will use a substrate which is changed into a colored product that can be viewed directly on the nitrocellulose membrane. If you are using a probe which is radio labeled as shown here, P32, then the nitrocellulose paper has to be exposed to X-ray film, where the process is called as autoradiography. And during autoradiography, the bands which are present or DNA of interest can be viewed clearly on the X-ray film. So this is called as southern blotting technique, where the DNA of interest can be viewed or obtained later, which can be used for the sequencing of DNA. So this is southern blotting. The next one is northern blotting. So northern blotting, as I mentioned, it is used for the identification of mRNA of interest or RNA fragment from entire RNA genome. So generally RNA genome containing viruses and all organisms can be used for the identification of RNA of interest. Here in this diagram it is shown that isolation of mRNA from the whole RNA extract. So here the entire RNA extract which is obtained from whole cell lysate, it is used for obtaining mRNA of interest. So now the RNA which is extracted is subjected to electrophoresis directly. Since RNA molecules are single stranded molecules, there is no need of denaturation step here, which is followed in southern blotting. So after electrophoresis in the agarose gel, then the bands on the agarose gel are transferred onto a special type of membrane called as DBM membrane, diisobenzyl oxy membrane or paper. So this type of DBM paper is used especially for mRNA because mRNA will bind strongly to this DBM paper. So after electrophoresis, the RNA fragments which are sorted on the agarose gel are transferred onto a DBM paper. So after transferring, the membrane is incubated with labeled probes. As I mentioned, it may be an enzyme labeled probe or a radio labeled probe. And the band of interest can be identified by viewing a color if it is enzyme labeled probe, or if it is a radio labeled probe, you can view the band on the autoradiograph. So by this technique, we can identify the mRNA of interest and you can collect it and you can use it for further studies. So this is northern blotting. So the next one is western blotting. So as I mentioned, western blotting is different from the other two blottings because the other two blottings are nucleic acids, whereas in western blotting you will identify the protein of interest. So the protein of interest can be identified from whole cell lysate protein. So what we have to do here is you have to collect the whole cell lysate protein. Then that particular whole cell lysate protein is loaded onto polyacrylamide gel electrophoresis. So sometimes here SDS is used as a detergent. After obtaining the whole cell lysate sample, it is treated with SDS, sodium dodecyl sulfate, a detergent which will give negative charge irrespective of the protein charge. So after treating the sample with SDS, it is loaded into polyacrylamide gels. So generally in southern blotting and northern blotting, we use agarose gels because the size of the nucleic acids is larger. So the porosity of agarose is also larger. So agarose is used for large molecular weight or molecular size compounds. Whereas the protein size is less when compared to nucleic acids. In western blotting, polyacrylamide gels are used. Since we are using SDS as a detergent for treating the sample, it is called as SDS page. So after sorting the proteins in the SDS page, the protein bands which are obtained in the SDS page membrane are to be transferred onto nitrocellulose membrane. So the gel is stacked with nitrocellulose membrane and allowed for electroblotting. So after electroblotting, the bands which are present on the nitrocellulose or our band of interest is identified with the help of labeled antibodies. So here directly labeled antibodies are used. Sometimes primary antibody and secondary labeled antibody is used. If the antibody is enzyme labeled, colored band can be directly viewed by using the substrate on the membrane. If the antibody is radio labeled, immediately X-ray it is subjected to X-ray film by the process called as autoradiography. So after autoradiography, on the autoradiogram, you can view your band of interest, protein band of interest on the autoradiograph. So this is Western blotting. So this technique can be widely used for the identification of viral antigens. Since antigens are almost 99% are proteins, you can use Western blotting for the identification of various antigens like HIV antigen, COVID antigen, and hepatitis antigens, etc. So all these antigens can be identified with the help of Western blotting. So in clinical laboratories, after ELISA, the confirmatory and accurate result test for any kind of antigen is Western blotting. So by this we can confirm the ELISA test strongly that it is the patient is having antigen. So this is the application of Western blotting in clinical use. So these are the three blotting techniques which are called as the heart of the molecular biology. So the next one is polymerase chain reaction. So this technique was introduced by Carey Mullis. Polymerase chain reaction. Previously, in olden days, for the molecular biology experiments, if the sample is less, it is difficult to conduct many experiments. For example, experiments that are associated with the DNA and RNA. Since they have negligible amount of sample, so it is difficult for them for carrying n number of experiments. After the invention of polymerase chain reaction, the sample molecules are amplified and you can perform as many as experiments as you can do. So the polymerase chain reaction is used especially for the amplification of DNA molecule n times. So sometimes it is used for the amplification of RNA molecules also. So but it is exclusively used for the amplification of DNA molecules. So while coming into the technique, so in PCR technique, the major components include your DNA sample, the DNA sample which has to be amplified, and the short primers and nucleotides which are also called as DNTPs, deoxynucleotide triphosphates, and a PCR tube, append of tube, and a buffer mixture, and an enzyme called as Taq polymerase. In general, the PCR is carried out in a machine called as thermal cycler. This thermal cycler maintains different temperatures required for the process of PCR. Since you can see the components on the left top, all these components are mixed and placed in the PCR tube, and the PCR tube is placed in the thermal cycler. So this is the process. So here each cycle of step, it is multiplied into 2 to the power of n times. So 1 DNA will become 2, 2 it will become 4, 4 will become 8, like that, 2 to the power of n. So while coming into the process of PCR, so it is a 3-step process, that is denaturation, annealing, and extinction. So generally for DNA replication, DNA polymerases are used. You can ask me a question, why here a Taq polymerase is used for the amplification of DNA. So in general, DNA polymerases are inactive beyond 40 degrees centigrade, whereas this Taq polymerase, which is isolated from thermophilic bacteria, Thermus aquaticus, is stable at 100 degrees centigrade, and it will show its activity in between 90 to 100 degrees centigrade. Another enzyme is used which can show its activity at more than 100 degrees centigrade, that is PFLU polymerase, PFLU polymerase, which is obtained from Pyrococcus fluorosus. In general, this tag polymerase or Pyrococcus fluorosus, these two enzymes are used in this process of polymerase chain reaction. You can see in the right side, so it's a three-step process: PCR, denature, annealing, and extinction. So all the three process are carried out at three different temperatures. Denaturation at 95 degrees centigrade, so that is for 30 seconds. Annealing at 55 degrees centigrade, that is 20 seconds. And extinction at 72 degrees centigrade, that is also for 30 to 40 seconds. So these are the three steps which are maintained at three different temperatures. So all these three different temperatures, 95 degrees centigrade, 55 degrees centigrade and 72 degrees centigrade, which are required for denaturing, annealing and extinction, are maintained exactly accurately in thermal cycler, or also called as PCR machine. So for the entire process, all the materials required for the PCR are mixed in the PCR tube, and the PCR tube is placed in the thermal cycler and allowed for all the three steps. So after completion of these three steps, you can obtain, and you can also set the PCR how many number of times you can amplify, that many number of times it will feed upon, and you have to use that much amount of the quantity of the reaction mixture for that particular number of times of amplification. So there are different types of variants like hot start PCR, real time PCR, etc. So whatever may be the PCR technique, but the basic process is this: denaturation, annealing and extinction, and the enzymes are tag polymerase and PLFE polymerase, DNTPs, and the machine that is used is PCR machine. So generally we can also quantify the amount of DNA amplified in PCR technique. So by real-time PCR, which is used for the quantification of the DNA amplification, and hot start PCR is also a variant of PCR where the tag polymerase is introduced in between annealing and extinction to prevent excessive temperature damage of the enzyme, which will take place in denaturation step. So like this, many variants of PCR are there. So where we can also quantify the amount of DNA amplification. With this PCR technique, after the invention of PCR technique, sample insufficiency is nullified and n number of times of DNA sample can be amplified. And this PCR technique is given by Carey Mullis. So the next technique is DNA fingerprinting. DNA fingerprinting is given by Alex Jeffries. Previously, before this technique, there is a problem. There is a problem for the identification of criminals at crime site, and also maternal court cases like baby mother issues, and also taxonomical studies are also difficult. After the invention of this DNA fingerprinting, all these techniques, construction of dendrograms, evolutionary techniques became easy, and also migration of species identification is also became easy with the help of this DNA fingerprinting technique. And this DNA fingerprinting technique is exclusively given by Alex Jeffries, and it is similar to southern blotting technique, where DNA isolation, treatment of with restriction enzymes, obtaining double stranded DNA fragments, conversion of double stranded DNA fragments to single stranded DNA fragments, and across gel electrophoresis, transfer onto nitrocellulose membrane, identification of band of interest. But here the difference is, for example, we will take a crime incident. A crime has been convicted, and suspected people are placed. So you want to identify the exactly accused person among the suspected people. So what you will do is, in the DNA fingerprinting is, it is as follows. So these are the three suspects. Suspect 1, you can see in the right side, suspect 1, suspect 2, suspect 3, and the DNA sample which is obtained in the crime region. So if you ask me the result, I can say from the crime scene, suspect 2 is the person who have committed the crime based on the DNA pattern. And how this DNA pattern is obtained is by this DNA fingerprinting. So what they have done is, you can watch in the left top, they have collected the DNA sample from the crime area. It may be a hair follicle or a nail, a piece of cigar, or any biological sample left by the convicted person. After collecting the sample from the crime region, if the sample is small, you subject the sample to PCR, polymerase chain reaction, which I have mentioned in the previous slides. Then you will obtain ample amount of DNA sample for the experiments. After PCR, you'll get the double stranded DNA n number of times. Now collect that, treat with restriction endonucleases. So similarly, simultaneously process the samples which are collected from three suspects. Suspect 1, 2, 3. Collect the DNA from them, collect blood. From the blood, isolate serum. From the serum, isolate WBC cells. Collect the DNA from them and subject it to restriction analysis. Now you have four samples: crime scene sample, suspect 1, suspect 2, suspect 3. So load all the four samples in four lanes in the agarose gel electrophoresis. After loading into agarose gel electrophoresis and subjecting the agarose gel electrophoresis, the DNA fragments are sorted based on their size. So larger will be on the top position and smaller will travel faster in the gel. It will be in the bottom of the gel. So after electrophoresis, transfer the DNA fragments onto nitrocellulose membrane and use probes. It may be radio labeled or enzyme labeled. If you are using enzyme labeled, you can use a substrate which is converted into colored product that can be identified on the paper itself. If you are using radio label probes, so in general, radio labeled, and these probes are collected from a special type of crate called as special type of crate. The name is not so important, and it is used for probing these all the four samples. So after probing, autoradiograph is developed by the technique called as autoradiography, as I mentioned earlier, and you will get bands on the photographic film as shown in this right side. So sample 1, crime scene, 1 suspect, 2 suspect, 3 suspect. From the above 4 samples, you can clearly see the band pattern at the crime scene and the band pattern of the suspect 2 are similar. So it shows clearly that the crime scene is convicted by suspect 2. Like this, you can identify children paternal issues by using DNA fingerprinting, and you can also study taxonomical. It can be used as a taxonomical aid for studying the migration of species and identification of species, and this technique became after its invention as a vital technique, and it is followed till today, though it is old, which is in 80s, that is 1986. It is still followed, and it will give still accurate results for the identification of criminals and paternal issues and some other types of biological studies. So once again, I thank you so much for patiently listening to my topic, today's topics, that is southern blotting, northern blotting, western blotting, polymerase chain reaction, and DNA fingerprinting. Thank you.